Good morning, y'all. Today is time to finish up our functional group notes. Just like we said before, I'm not going to take too much for this part because you know what it is. Anything that is not hydrogen that's stuck to a hydrocarbon, we call it a functional group, and that's where all the fun, weird stuff in organic chemistry comes from. So let's just get at it and start up with this guy, the organic acids. Now look, here's the deal. There are also bajillions of kinds of organic acids. Like the one that everybody's most familiar with, don't even know it's an acid, is DNA, deoxyribonucleic acid. Now, why is this molecule that is like a meter long called an acid? Because at the very tail end, there is this. There is a carbon with two double bonded oxygens and a hydroxyl group all on the same carbon, right? And so this R, in this case, this little R right here, that represents, like over here, it's like the rest of the DNA molecule. But there's this right here, okay? And what happens in an organic acid is this hydrogen. Oops, hold on, guys. That hydrogen right there, like breaks off, whoop, and goes into solution, and you're left with this. And so that's how it becomes an acid. The thing that you need to look for in a chemical equation or in a chemical formula is this: COO. Anytime you see COOH, just know. Just be like, COO. That's an acid, man. Just be like, you know it. This is right there. And there's so like in that condensed structural formula, see, it's like CH3, CH2. Usually it'll be written like this: CH3, CH2, COOH. That's how this is usually written. But in the reference table, they're writing it like that, just so that you're not like fooled too easy. All right, so why is it an acid? Because this hydrogen right here breaks off into solution and forms a hydrogen ion. Therefore, it's an acid. So they are weak electrolytes. They're very, very weak, right? So because what happens is that this hydrogen breaks off, and any other hydrogen in there, it pops right back on. So these don't break apart terribly often. I think it's like 30% of the time that these hydrogens are actually broken off and acting like a hydrogen ion. What is it? 70%? 7, 8, 9? Yeah, I guess the remaining 70% of the time it is not broken off, so you don't have an acid. Do you guys get what I'm saying? So let me take a step back. The organic acid, the functional group looks like this, right? Where you have, what's it called? A carboxyl. There's your carboxyl group, and a hydroxyl group right there. This is the whole thing, and this is what the name looks like. What we're gonna do is we're gonna change the end to oic and then say acid. So just as before, you count the longest sequence of carbons: 1, 2, 3, meth, eth, prop. Instead of propane, it's propanoic acid. That's really all there is to these things. Like these guys are simple, right? Oh yeah, change the ending to oic acid. So for this one, here we have, let's do Viking red here, guys. Here we have 1, so that's methane. But instead of the E, we're going to put oic, methanoic acid. Yeah, see how that works, right? Yeah, not too hard. This one, 1, 2, oh, okay, first thing we should do is circle and identify our functional group. Definitely an organic acid, because we got the double bonded oxygen and the hydroxyl group. So then we just count up our carbons: meth, eth, prop, oic, acid. Lovely. These acids are usually pretty benign. We actually have evolved quite a taste for these things. Most organic acids are going to taste quite delicious. Like almost anything citrus, it's like that citrus acid tastes so good. Vitamin C, it's an acid. It's a lot of that stuff. It's actually quite yummy. Vinegar, it's an acid. It's actually quite yummy. All right, so there's structure to name. Here's a condensed molecular structure. It's just Q. Man, it's just the straight acid part. So, because we see COOH, that's how we know it's going to be an organic acid. Why? Because that's how we write it, so that we know that it's an organic acid. And if there's only one carbon, then it's going to be methane. Oh, look, it's this one. Same thing, just written completely different. Right? So this is the difference between, this is your structural formula, and this is your condensed structural formula. Same thing, just presented to you differently. All right, this is all right. Okay. Actually, this shouldn't be too bad. This shouldn't be too bad. Because look, to name this thing, I see right here, that's how I know it's going to be an acid. So all I need to do, count up the carbons, call it oic acid, right? So let's go. Meth, eth, prop, wait, meth, eth, prop, bute, meth, eth, prop, bute, pent, hex, hexane. Oh, sorry. Well, hold on a second. I kind of goofed that one up there. It's hexane. Oops. There you go. Hexane oic acid. And we're going to do that multicolored just because that's how we roll today. Next one. You guys see how this works? Naming these ones shouldn't be hard. Look, COOH. You know it's an acid. Meth, eth, oic acid. Not too hard, is it? What you need to be able to do here, my friends, is you need to be able to look at the structure. You need to be able to like recognize the functional group. I need you to be able to see the functional group, circle the functional group, and then know that it's an organic acid, right? Also, if it's written out like this in condensed formula, I want you to recognize the functional group and then name it. That's all you have to do with these. That's really like the beginning and end of it. Propanoic acid. That's easy, man. We can draw it. Prop. Meth, eth, prop. That's 3. Meth, eth, prop. Now it's an acid. So what's the functional group for an acid? Well, we're going to come back up to your reference charts and see right here. Yeah? A double bonded oxygen and a hydroxyl group. So how about a double bonded oxygen, hydroxyl, and then fill everything up with hydrogens. All right, that's easy. That was easy. Vinegar is an organic acid with 2 carbon atoms. What is the IUPAC name for vinegar? Oh, this is also another question that comes up a lot. What does IUPAC mean? This is the International something physical something chemistry. International Union of Physical and Applied. I think it's the International Union of Physical and Applied Chemistry. Something like that. It's the international governing body of chemistry so that everybody's doing science the same way. Right? So we all came together to figure out how to name things the same way because there's like 7 or 8 different names for everything. Vinegar, sometimes it's called vinegar. Sometimes it's also called acetic acid. Sometimes it's called glacial acetic acid. So everyone's like, this is stupid. Let's just come up with a uniform way so we know what it is in the name, which was brilliant. So, 2 carbons. Oh, wait, that's F. Eth, but instead of ethane, ethanoic. And then we just say acid. And now this one wants to know what's the structural formula for vinegar. Well, if it's only got 2 carbons, 1, 2, OH, double bonded O, right? And then we throw in hydrogens. Voila! It's so easy. We can do organic acids here, guys. I can sit here and do organic acids all day, but I'm not. You know why? Because we're going to enter into what is, in my opinion, the most difficult one yet, but also the most interesting and awesome ones. These are the esters, which also sounds like the name of some old ladies who sit like English ladies sipping tea, like with Esther. Esther and Ethan. Esther and Ethel. So here's the deal. Esther and Ethel, they're like the old lady mascots of organic chemistry. So like here's the deal. These things, these esters, these are actually super awesome. If you've ever smelled anything or like tasted like a lollipop or any other flavoring, it's probably some kind of ester. These things smell amazing, and also they smell just like puke. So the Betty Bott's Beans of Discipline, right, where you have to taste the flavor of your misbehavior, which is often like, you know, dog food and vomit and earwax. The way they do that is with esters. They are, so it's not actual earwax. It's not actual vomit. I think there are laws against like feeding people earwax and vomit. But we can make an ester that smells and tastes just like vomit. It's amazing what you can do with these things. And what it is a combination of an alcohol and an acid. This is like another reason why if you ever hear like wine people talking about wine, they'll like swirl it and be like, oh, it has the bouquet of smoked cherries and apple blossoms and vanilla bean with like, I don't know, they just go off on it. Well, the reason why things that are fermented can have like really weird like flavors and whatnot is because during fermentation, they're like, you know, fermentation makes alcohol. But at the same time, there are all these organic acids from the grapes, from the grape skins, and from the yeast and all the stuff that's in there. And all you need to do to make an ester is an alcohol and an acid need to bump into each other. And if they bump into each other, bam, you get yourself an ester. And so, if this is done well, you can get like wines that taste fruity and floral. Like really, all it is grape juice and alcohol. But because of all those acids that are in the grape skin, it's like you get all these other weird flavors. You get it? Okay, so here's the thing, is when you look at an ester, it's going to be like a carbon, a hydrocarbon on that side, a hydrocarbon on this side, and in the middle, you have what looks kind of like an organic acid. You have a carbon with a double bonded oxygen and a single oxygen, but instead of being on the end, right, like for an acid, it's on the end, so the acid looks like this. But for an ester, that hydrogen is gone and it's replaced by a link of carbons. You got it? And the way you make it stinky is over here, like this, all you need to do is change up the R groups that you have. So like, the way that you change the stink flavor from like, I don't know, butterscotch to vomit is you just change the ratio, like this one, and those combinations of those two R groups is what gives it its unique flavor or smell, which is unbelievable. You just change the number of carbons and it goes from pineapple to rum to vomit. It's amazing. Throw it in a lollipop and man, there you go. Puke flavored lollipops. And this is what it looks like, right? A bunch of carbons, double bonded oxygen, and the oxygen just kind of in the middle, and then some more carbons after it. And so naming this thing can be a little bit complicated, all right? So there's going to be what we have a root name, just as always, right? You have like the that base root name, and that always goes over here on this side to start with, and then we have an alkyl group. Well, so here's the deal. The group with the oxygens is going to be the root name, right? And it's going to end with O8. And then the other group, or like the other R, like this guy over here, that's going to be your alkyl group. So this is your root. And this is the alkyl. So I said here the group with the oxygens. So the way that we're going to do this, like the way that you can tell which is the root, which is the alkyl group, is you take that oxygen right there, and we're going to cut this thing right down the oxygen. Okay? Because that's sort of like a nice middle indicator. It's like an oxygen. Right, so look on one side, look on the other. The side that has the double bonded oxygen, right? So if you have this, that's how you know it's the root name. Got it? We'll do this a couple times. I think it'll make a little more sense to you. Oh, how about right now? So here we go. We've got some carbon, 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 oxygen, carbon, carbon, carbon. Oh, and like a double bonded oxygen. So here, that is my functional group. You see what I'm saying? So what I'm going to do is I'm going to cut this thing right down the oxygen. And this is the side with the oxygen. So that is my root name, and it's going to go over here. It's going to go first. So meth, eth, prop, bute. Butane. but instead of an E, it's going to be O8. Now, how did I know it's going to end with O8? Because back up here, the ending is O8. Right? That's how we're going to use the reference chart to remember the endings. Remember, we're not memorizing, we're using the charts. That's how this is supposed to work. So that's this, butanoate. That's the root. And now this is going to be my alkyl group. So what's the name of an alkyl group? Remember, they end in O or YL with one carbon. Yeah, methyl. Methyl butanoate. Easy, right? Quick review, because it's so easy. What we're going to do is we're going to look at the structure, right? And the first thing we're going to do is cut it in half right along the oxygen. The side that has the double bonded oxygen, that's going to be our root, and the side without the oxygen is going to be our alkyl group. Got it? Oh, also, fun fact, the reason why we call this the root is that this side with the double bonded oxygen, this used to be the the acid. The side with the double bonded oxygen used to be the acid, and the alkyl group is the one that used to be the alcohol. How do you know? Well, acids are the ones with the double bonded oxygens, not the alcohols. Yeah. All right. Let's okay, one more. All right. So here's our functional group right here. All right, carbon, oxygen. Okay. So I'm gonna split it right down the middle. And this is the side with my double bonded oxygen. So this is going to be my root. And this will be my alkyl. That's a terrible A. So the root has 1. So it'll be methane. Oh wait. How do I end these? Oh yeah. O8. I'm going to end it in O8. So it's not going to be methane. It's going to be methane. O8, not O, but O8. Okay, so methanoate, and my alkyl group, ethyl. There's our organic chemistry mascot again. Ethyl, methanoate. Good? You want to try another one? Too bad, we're doing it anyway. Why would you want to? That'd be weird if you did. Nobody wants to do this. Yes, we do. There's people that do it for a living. All right, once again. Carbon. Ooh, there's our functional group. That's our ester. And okay, so and there's my oxygen. So I'm going to split it right down the oxygen. This is going to be my root because it has the double bonded oxygen. The double bonded oxygen, this used to be the acid. And here is my alkyl group. No oxygen, so that used to be the alcohol. So ethyl or ethane. Switching it to O8, and my alkyl group, ethyl. Ethylethanoate. Say that 10 times fast. Ethylethanoate. 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 So ethylethanoate probably smells delicious, or who knows? Maybe it smells like wildflowers or plums or lilacs. I don't know. Or it could smell like dog poop. These things are amazing. Really. If you added like this molecule right here, it could literally smell like dog poop. You add like 2 more carbons, and all of a sudden it'll smell like roses. It's like these things are just, they're actually fun to play with. I'm really bummed we can't do a lab with these because, boy, it smells so bad. Like the whole room will smell like Betty Bott's beans. It's just vomitous, man. It's terrible. Some things are really maybe not that bad to me. Moving right along to our next is going to be this one, and we're going to look for our function. Oh, look, an ester functional group, right? Double bonded oxygen with an oxygen in the middle. So I know how to name this. First thing I'm going to do is split it in half. This is the side with the double bonded oxygen. So this is from the acid, and this will be my root name, right? Because my root name comes from the acid because it has a double bonded oxygen. And this is going to be my alkyl group. So the root name, ethyl on the ethanoate, and this is going to be methyl ethanoate. Big, awesome. Remember, the side with the double bonded oxygen comes from the acid, and this side comes from the alcohol. That's it. I think that's all you need to know about it. Yep, that is it. That's all I need to know about that one. All right, now these last two we're going to talk about, these are not terribly complicated. The amines, really, an amine is just a nitrogen that is stuck onto a hydrocarbon, and that's where, nitrogen. That is where some crazy chemistry starts, because no proteins can happen without nitrogen. That's why nitrogen is so like deadly important to us in our agriculture and our general health. You might not know it, but this little element right there is like one of the biggest fundamental problems that humanity had to overcome for our civilization to be where it's at. Haber did it, and then he killed unbelievable numbers of people. But anyway, that's a whole nother story. The amines, this is just a nitrogen attached to a hydrocarbon, and it's going to be very much the same. Root, what, where. Butane amine is just going to end, let me come back up here. It's going to be a little different, as in, because the ending is just going to say amine. And then we need to say where. Right? But that's it. It is a little bit weird because it's an amine. It doesn't follow the exact same pattern. So it just ends as amine. Right? So here, meth, eth, prop, but. You take butane and then you change the ending to amine. And then indicate where. You understand what I'm saying? So how would we name this mess? Why would I do this? All right, I'll tell you what we do. We're going to start, first I'm going to try to find the longest consecutive sequence of carbons. And so I've got 1, 2, 3, not a carbon, not a carbon, not a carbon. Okay, over here. Not a carbon. All right, okay, that's it. Okay, so what if they give you a weird looking molecule like this, which often happens, like molecules are not often drawn for you to like feel comfortable. So I'm going to draw it, and for me, I'm going to circle the carbons, and then I'm going to start numbering, because I think the important thing that I need to keep in mind is that this is my functional group, right? That's like the thing that is not hydrogen or carbon. This is my amine group. So I'm going to start counting from this side and number this 1, 2, 3, 4, 5. Okay, so that gives this number 3, and I'm going to number this way. 1, 2, 3, 4, 5. Looks like it doesn't matter at all, like which direction I start naming. But what's nice is I know I have 5 carbons, so that's going to be pentane. But instead of the E, I'm going to have amine. Wait, is it amine? How do I end it? Oh yeah, pentane. Yep, just amine right there. And then I got to indicate where it is. So that's on my number 3 carbon. 3-pentanamine. That's an ugly, ugly molecule. Not gonna lie. I don't like that one. Alright, and the last one, amino acids. So for an amino acid, what we need you to do is to just recognize that you're looking at an amino acid. You don't need to name it, nothing. That's it. And the way that we know that it's an amino acid is there is an acid and an amine, all attached to the same carbon. So you look at it, you say, oh, here's an amine, and oh, there's an acid. Do you see how that name happened? There's an amine. Oh, there's also an acid. An amine. Oh, acid. Get it. I don't think that's really how it happened, but that's how I like to think of it in my brain. So that's how it happened, as far as I'm concerned. So yeah, an amino acid. You have an amine on one side. You have an acid on the other. And then this R, this is the part that like all you weightlifters and like muscle heads want to like pay attention to, or also anybody who's into health and nutrition. This R, this side chain R, is what determines which amino acid it is. And for the amino acids that we eat to be like, you know, healthy and strong to make our proteins, these are what make the different amino acids. The shape of this side chain is what determines which amino acid it is. And then this thing, right? This is what actually helps them link together to make those long chains that fold up together because of hydrogen bonds to make our proteins. Do you understand what I'm saying? So this amino acid part, this here, the amine and then the acid, that's the linkage. This is the thing that is used to put the amino acids together. So if you remember back in Krekel's class when you were learning about the production of proteins, the ribosomes hold the amino acid in place, and so the next amino acid comes in, they link together. The next amino acid comes in, they link together. The next amino acid comes in, they link together. And that is how, like, the RNA is transcribed into a protein, right? And the way that those proteins link together is this big mess right here, okay? So what you need to be able to do is, like, recognize it. Recognize that it's an amine and then an acid. And when they link together, they make this thing called an amide. And here's how it happens. Here's how it happens. I want you to like lock in here. You see how this is your amine, right? And then right here, that is your acid, right? So this is our amino acid, and here is our other amino acid. What color is not in there? How about purple? So here's our groups. We have our amine, and here we have our acid of one, and here's our amine, and here's the acid for the other. So the way this happens is the ribosomes hold these together just like this. So it goes nitrogen, acid, or I'm sorry, amine, acid, amine, acid, amine, acid. And the job of the ribosomes is just to hold these two things together. And when that happens, when you have an organic acid right next to an amine, look, you get two hydrogens and an oxygen right next to each other. But so can you think of any molecule at all made of two hydrogens and one oxygen that is incredibly, incredibly stable? I'll give you a minute to think about it. You're right, water. So what happens is these just get close to each other and they break off and form water. You dig what I'm saying there? Why? Because two hydrogens and one oxygen make one of the most stable molecules ever, and if it's stable, it's going to happen. Okay, and so what that does is this bond right here is left open, and this bond right here is left open. But they're not just going to, like, the electrons aren't just going to be wiggling around out in outer space. They're going to like link together and make a covalent bond. And so this bond right here and this bond right here come together to form this bond. With the help of the ribosomes, sort of holding them right in place, so that the spatial orientation is just right. All they got to do is be near each other, and wham, bam, they're done. And that is how you make, like, a protein. And this functional group right here and this functional group right here are what identifies those proteins, or those amino acids. Link them all together, proteins start to fold. That's how you make a red blood cell, or I don't know, muscle, or piece of connective tissue, or your fingernail. Man, I don't know. Whatever your body's doing that in. You got it, and we call these amides. We don't call them polypeptide bonds, because what the heck is a polypeptide bond? Those biology people are out of their mind. So an amide is this. It's a bond formed by the combination of two amino acids, where the acid and the amine stick together. Can I make any more circles here to make this any possibly more, like, clear to help you understand what I'm talking about? Yeah. So also, this is called the condensation reaction. Why? Because we're producing water. Here's more drawing for you. And what do you say we just stop there for today? Right now. Have an awesome day. Hope you guys are enjoying some of the nice weather.